we will go ahead and get started. I see that there's a handful of people listening live right now on Zoom. That's great. So today for our monthly CSO webinar series for the KdVS Foundation, I've asked one of our leading researchers, Dr. Yann Herault, who is a scientist or a neuroscientist at the Institute for Genetics and Molecular and Cellular Biology. You'll notice I didn't try and pronounce that in French, obviously, where it says in Strasbourg, France. And Dr. Herault is the researcher who established basically the only, that I know of, rodent model for Koolen-de syndrome. And so for that reason, I asked him to come and speak today to sort of give you an overview of his work, how rodent models like this really are critical for having tools to develop treatments in KdVS children and young adults. And also, Dr. Herault has received one of the KdVS investigator grants, and so has a couple new exciting projects that he's working on in this space. So without further ado, I think when I clicked some buttons, I made it so that you could share your screen, but go ahead and double check. Yeah.
Yes, I will do.
Okay.
Okay, thank you very much, Anna, for the kind and invitation and the great present and the great introduction. So I don't know which one of my screen are you looking at? You are looking at the good one with the slide presentation, or
I am looking at, like, it's not a full screen right now. I see all of the
okay.
slides on the.
I start again. Sorry for that.
That's okay.
Yes, this is this one. I have too many screens, so this is this one. Okay,
yep, beautiful. Looks great.
So it's a great pleasure to be here. As Anna told you, I'm a neuroscientist, a neurobiologist, and I'm working in Strasbourg. So I just have a slide to show you where Strasbourg is. So Strasbourg is located in Europe, you know, in this just close between the French and the German border. And it is very nice place, you know. You have these very beautiful houses which has been settled in the 16th, 17th century in France. And I'm working in this institute. I don't know if you can see okay, I will put my pointer laser here. And I'm working at the IGBMC. You can see the difference between the two types of buildings. So, and this is a building which is quite unique in France because it was built by a US architect, in fact. So this is not usually the type of building that we have even for research in France. But okay, saying that, I've been investigating, I've been interested by the Koolen-de syndrome because I met more than 14 years, 15 years ago, Bert de Vries and David Koolen, who were just investigating this new syndrome that they discovered. And we discussed, because we had tools at that time to manipulate the genome in order to generate similar defects that you can see in individuals with KdVS. We decided to go further into that and to mimic those changes in the mouse. And why we did that in the mouse is because the mouse model has been used in biology for many years. So it started by a French, in fact, professor, Lucien Cuneau, who showed that the Mendel's laws that are driving the transmission of genetic traits are also valid for mammals. And he did that using the mouse. And then there was a number of investigators based in US, like William Castle and Clarence Little, or who took the mice as a model for investigating already at that time cancer and other type of disease. And they did that with people like Abilatrop, who provide us some mouse model. And this has started the story because those two people, for example, William Castle and Clarence Little, they made the first inbred strains. So these are mice which are homogeneous in terms of alleles, so they are quite similar to each other's. And then later on, there was three key people, Mario Capecchi, also based in the US, Martin Evans and Oliver Smithies, one in UK, the other is also in US, who set up the capabilities of manipulating embryonic stem cells using the mouse as a model system. And this had changed the way we could generate change in the genome of those animal models. And saying that, after the main, let's say the main development were, first of all, the sequencing of the human genome, which was done in 2000, then followed by the mouse in 2002. And then there are also new tools which has been made, which allow us to modify the genome in almost every species we would like to do that. And this, for the mouse, has led to a certain number of high level of publication, which has increased dramatically even compared to the rat model, which is also another type of model. So this is the number of publications that has been put in a very well-known resource for publication, which is a PubMed, and this is a number of rat publications. So this is just to give you a landscape about the use of the model in research. So why we are investigating KdVS in the mouse? It's because first, the region, which is indeed modified in people with KdVS, is similar, really found in the mouse, but in another chromosome. It's not exactly the same context, but you can appreciate that all those traits here, which are in fact the representation of genes in the genome, like cancel1 here, we'll come back to this one in particular, is also located in the similar region in the mouse genome, close to MAPT, which is another gene found in the region which is deleted in individuals with KdVS. And this is the same for CHRR1, which is another gene also found close here. So relatively the regions with the same genes are located in the mouse genome. So there is a good homology, this is what we say. So it's quite well preserved. So saying that, we could now, with certain type of genetic tools that I have maybe a time to discuss further, we could go and make a model, an animal model, a mouse model, to better understand the Koolen-de syndrome. And this was indeed the goal. First, to get a better knowledge on the genotype and phenotype relationship, although we get information from human, but here we have another way to achieve that, and also not only to look at adult or young stage, but also to go during development. We can do that also to understand better what are those changes are inducing other changes into the genome and into the cells. So what we call the deregulated pathways, and what are the key genes where when we have one of them lost are causing this syndrome. So these are dosage sensitive genes. And of course, the ultimate goal will be to carry out therapeutical intervention in those clinical models. That could be the first step before going into human, to alleviate or to improve the welfare and the phenotype observed in those models. But of course, at first, we should make the model. So I'm not going to detail that too much, but because this has quite a genetic construct, this is more for, let's say, students or for people who knows. So we are using a tool that is maybe different from what you have heard before, that allows us really to target precisely into the genome, the region of interest. And having those tools inserted here upstream of the region homologous to the KdVS genetic interval in the mouse, and downstream we can generate by a combination of with another combination of genetic tools, we can generate the animals that will carry the deletion of that region and other animals that will carry the duplication of that region. So the duplication is not really interested for the Koolen-de syndrome, but it's very interested in terms of genetics, and I will show you why. Because we can now compare the normal population, we have 2 copies of the regions of the Koolen-de region, with those which have lost 1 copy, which is similar to what is observed in some individuals with KdVS. And we can also compare with those where will be restored the 2 copy in a different way. In one chromosome, there will be the loss of the region, but on the other chromosomes, there will be the duplication. So you will have, overall, you will have 2 copies, okay? And although it's a bit different, and also the case of there is a duplication with 3 copies, which could be a side effect that we need to investigate. So we were able to generate those animals and to identify them relatively easily with a genotyping assay that allow us to discriminate individuals that carry the deletion with this type of band, for example, from those that carry the duplication with this type of band. And of course, when they are del and dupe, they get the 2 bands. And when they are not, when they are controlled populations, they don't get these bands, but they get others, okay, that you can find. So it's a bit complicated, but this is how we work. Because we should be we should very well identify and very well precisely identify those different type of genotypes. So we use those different genetic configuration in order to see what could be the consequences of those genetic modification in the mouse. So of course we consider what was observed in people with KdVS, and we focus first our attention on cognition and behavior. And you may not know, but it's possible using the mouse model to evaluate different type of cognition traits and behavior traits by using different type of tests of paradigm, which has been validated by the scientific community. And I will introduce you some of them. Some are better for monitoring, for example, the locomotor activity, others to evaluate the anxiety of those individuals, of those mice, and also others to go for learning and memory, to test their learning capabilities and how long they will keep a memory of one of a series of events. So this is what I'm going to introduce you. I don't have time to present everything, but this is what we did for the whole story. So for example, there is one very easy test, a nice test, which is not really which is only based on the curiosity of the animals. So we allow the mice to explore an open field, which is a cycle 1 or square 1, okay, for 1 day before the first phase of the test. Then we do place in this open field the second day 2 objects which are identical. So here it's 2 dice, but it could be 2 different types of objects. And the mice will be able to freely explore those 2 objects for 15 minutes. Then we can use a time for challenging the mice with a new object. And here, so the mice is put back to the cages where she is living, and then she went back to the same open field in which we have now moved one object to a new one. So here it's I don't find the English term. Oh my gosh. It's another object. Okay, so now usually the wild-type mice, what they will be doing is that they will be spending more time exploring this new object compared to the dice. And so this is what she will do. And if you score the time they spent exploring the new object versus the familiar one, you will be able to have a kind of discrimination index where you score the time exploring the new object versus the time exploring the new versus the familiar object. And indeed, you can see that the wild-type mice, they spend more time here doing that. And for the DEL carriers, so those which mimics the KdVS condition, we have a deficit compared to the one-time. So it seems that their object recognition memory is not as performant as the wild-type is. So this is what we did, and whereas those 2 other genotypes, they are not affected here. And we have a compensation in the del dupe, which is what we were expecting, because here too, we have 2 doses of this functional region. So we did also other tests with a spatial memory. So it's another type of test. I can explain it to you very briefly. So we have a kind of small swimming pool. We call it a Morris water maze for the mice, because it was designed by Morris, a scientist, a UK scientist, in which he noticed that if you place just below the surface, which is opaque for the mice, a small platform, you can make the mice learn to find where the platform is located. And she will be using for that cues, which are located outside of this water maze, which are on the wall, in fact, of the room. And she will, by having different sessions every day, the mice will learn very easily to find where the platform is located. And this is based on spatial memory, because as you can see here, for all the genotypes that we have analyzed, so I will be very brief on that, all the genotypes perform very nicely. They learn after 1, 2, 3, 4, or 5 days. They start to be a plateau here, where they travel the same distance to reach the platform. And they can remember also very well where the platform is located. And we did that 2 different times, and the memory is not impaired. So the spatial memory doesn't seem to be affected in those mouse models. What we did also is we look at other changes which has been observed in people with KdVS. We look at craniofacial features. And to do that in the mouse, at that time, we were using landmarks. So these are these red dots which are located on the skull, on these skull figures. And we are making X-ray scan, 3D X-ray scan of the cranium of the mice. And we can put those landmarks, which are precisely located at for different parts of the skull. And using, let's say, using Euclidean distance matrix, so we can calculate the distance between the 2 landmarks, we can see the changes which are appearing in those animal models. And this is what we did. And just to summarize, using those type of landmarks and those distances, we can easily discriminate the group of animals which are carrying the deletion from those which are the control population, and also from those which are carrying the duplication. And this can be done with different type of analysis. I'm not going to detail that too much. It's a principal component analysis. And you can appreciate here the change that you can observe in the mutant population here, for example. So in this axis, this is an increase, in fact, of the size of the skull compared to the reference population, which is observed. And you can score better what are the changes associated to this population with this genetic alteration compared to the control, which is quite similar to what is observed in people. What we did also, we can look at the brain architecture, and we did that with a nice team which was located in Toronto at that time. And we did MRI scanning of the brain of those mutant mice, of those different genotypes. And we didn't find strong differences in the volume of the brain of the deletion carrier, for example, here compared to the control population, the wild type or the del dupe, but the volume was slightly decreased in the dot plus. But we were keen on seeing, so these are for the whole size of the brain. But now if we look more closely to regions which are also known to be involved, for example, in the novel object recognition or into some of those deficits that we found, we didn't find any changes, for example, in the hippocampus here, which is also a place very important for the mice to learn where the place was located. But we can see that, for example, here in the entorhinal cortex, which is a very specific region of the brain involved in this discrimination of the novel object, the size of this region has been affected. So we need to understand now how this change have happened, what are cells and molecular, the cellular subtype which are affected. These are things which could be done. So what we did also, because we knew from Bert and from David that those people with KdVS have a change in their social interactions. They are more friendly. This is what they told us. So we tested that using a paradigm which was developed by Jackie Crowley in US. And in those paradigms, you have 3 steps. So for the first 10 minutes, the mice are placed in this arena where you have 2 rooms, where there is a door for each room communicating here, and you have 2 cages which are gridded cages. So at the start, those cages are empty, and you place the mice in the middle of this nest. So then you have to let the mice habituate to this new environment. Then after you put them back to their cages, and they come back to this new session where you have one individual here that will start to be the first individual that they will meet. They have not met these mice before, and we will score the time spent by those mice looking at the new familiar, so the new individual, which is located here, and we can compare with the empty cage. And what we have been able to see is that during those sessions, the first session, but also the second session, I will show it what it is. The time spending sniffing the new congener. This is the way the mice start to interact here in this type of paradigm is increased when they are carrying the deletion of this region. So it's likely similar to what is observed with the friendly behavior observed in individuals with KdVS. But so there is another step of this of this paradigm, which is due after another session, session 2, in which now we are putting a new congener on this part, and thus this will be the familiar congener. And usually the mice spend more times according to looking at the new congener compared to the familiar one. It's like for the object. And indeed, we had this similar behavior. So if we score the sniffing time with the familiar or the novel individual, you can see that there is an increased interest for the novelty, for the novel individual, and this has been seen for all the genotypes. So there is no defect in social preferences. So this is quite important because, for example, in autistic models, this is completely disrupted. So this was fitting quite well with what we observed in human, and we did also a final test in which now we have two animals which are freely moving in a small environment, and we score the time the mice spent by interacting together. And this was quite basic test, but here too, we were seeing that those individuals carrying the deletions, they were spending more time with social interactions than the other genotypes. Okay. So if we look at the scores that we have done, also including other type of tests that I have no time really to present here, we were scoring a certain number of defects, which was really significantly affected in the deletion carrier, which was quite similar to what has been observed in individuals with KdVS. So I didn't have time to show you, but there is a decreased activity, like similar to hypotonia. There is some alteration of the memory, especially in the recognition memory. There are also some repetitive behaviors that we have observed in some individuals, it's not all the population. And there is this friendly amiable phenotype that we have scored, which is frequently associated with people with KdVS. So this is what we were stating that, and we published that this model were recreating the most frequent defect observed in human at that time. There was no effect, so there was a slight things that I have no time to discuss, and there was also an effect on the body size, but which was seen in all the different genotypes, so that I have no time really to compare. Okay, so what we did after that is that we follow the research of Bert. We were discussing, of course, with them during the process of the analysis of this mouse model, and they published that cancel-1 mutation can also drive similar consequences and could be part of the Koolen-de. So cancel-1 is one of those genes which is located in this region. It's preserved. It's also found in the mouse. You can see it in human here. So these are different lines because it represents different ways the gene is expressed, let's say. And you have similar things in the mouse. And what we decide to do is to look at the consequences of inactivating one copy of cancel-1 to see if it could mimic what's the kind of mutation which is observed in individuals with KdVS. So this is what we did. So but before that, just to introduce you cancel-1. So it's a very interesting gene. It's a transcription. It belongs to the proteins that are going to modify, in fact, the expression of many other genes into the genomes. It belongs to a complex which is called NSL for NSL, let's put it this way. It's not really important. It's integrated in this complex and contribute, in fact, to the function of this complex. And so what we decide to go is to look at mutation. We cannot, at that time when we did this experiment, we were not able to make point mutation as it was described in this paper by David Koolen. But what we did is we use a resource that has been developed by the scientific community, the International Mouse Phenotyping Consortium. And at the moment, I'm chairing this consortium, which is a global consortium aiming at getting the function of all the protein coding genes in the mouse genome. So we take advantage of this consortium in order to recover this allele of cancel1, which is in fact a knockout allele of cancel1. So this is what we did. And we compared in the same battery of tests, we compare the effect of the cancel1 mutation, the cancel1 loss of function in only one copy. So it mimics what is observed in the deletion. And to see whether or not it can reflect what we were observing in the deletion. And you can see with the color here that almost all the phenotypes that we have scored in this deletion carrier were also found again in cancel1 with some slight differences. For example, there was a lower activity of exploration of a new environment in the open field compared to the deletion, but that's slightly differences. And the social interaction, we were not able to score again in this mutant. So there may be slight differences, at least in the mouse. For the moment, I cannot state that. So I think as a conclusion, the cancel1 heterozygote mouse model recapitulates most of the defects which are observed in the KdVS model and also in human. There is no impact on the social interaction, so we don't know exactly where does it come from. There is maybe another gene in the region, which is in fact playing this role. And for the moment, we don't know which one it is. And they have a stronger phenotype because they are also affecting the rotarod, which is a test that is used to look for the coordination of the animals. So yeah, so it's more about the locomotor activity and the movement. So we went a bit further with the help of two people later on to characterize further the social behavior in the KdVS model. And this was done with the help of Fabrice de Chaumont and Elodie sorry, she's now in my research group in Strasbourg, and she developed when she was in Paris in the laboratory of Thomas Bourgeron, a nice way in order to look at the social interaction of mice in an environment which is quite big, and you can follow the interaction of up to 4 mice at the moment and to see what they are doing together during a long period of time. And so we implement that in Strasbourg, and we were keen on looking at different type of behavior. It's much more detailed than what we have done before, so it's much more precise and interesting. So this is what we did, and we did that with 4 individuals, which are two individuals from the control population and 2 individuals that carry the deletion, which are kept together, which know well each other. And what we are able to see is that there is a change in certain type of phenotypes. There is a decrease of all the time the animals are spending alone, moving alone or stopped alone. This is shown here. Is highly significant, while there is an increase in all the social configuration event, including group of 2 mice, group of 3 mice when they are moving side by side the same way, or when they are on the opposite way, when they are doing contacts nose to nose, or when they are doing contact nose to genital, which is something which is, as you know, if you have a cat or a dog, this is one way for the animals to recognize each other. So, and what has been also found reduced is the number of breaking contacts. So when they are not together. So which is quite interesting because it's much more detailed to what we have published before. So this has not been published, but this is very relevant, I think, to the human traits. And it's very interesting if you want to understand what is the basis of being friendly, for example, in the normal population. So for the conclusion and perspective, so we have good, I think we have we have good basis to say that KdVS is probably due, in fact, to the loss of cancel-1, but we should not forget that there are other genes in the region that could contribute to some of the KdVS features. We are keen on going further in investigating the role of cancel-1 and certain modification that allows the expression of genes during neurogenesis and brain function. I think it would be important to understand that for future treatment. To determine also what kind of brain cells are altered in KdVS. Is it during development, for example, during the setting of the brain structures, or is it during the functioning of those neurons or those cells which are supporting the growth or the function of neuron, like astrocyte, for example? And of course, we might also think of building a new models for more valid therapeutic evaluation. And this is something that, and of course, there is also maybe other interest for this region, because this region has been, the duplication of this region has been implicated in neurodegenerative disease. So this is another aspect of the region, which is not, at least for the moment, at least considered for the deletion, only for the duplication. But saying that, as a perspective, I think I would like to answer maybe two main questions. The first one is how far point mutation, which has been described in KdVS, I made a mistake here, KdVS individuals cause similar defect to the cancel-1 knockout. Because this we don't really know. A point mutation can alter the function of a protein in a different way than causing the complete abnormal function of the protein. And can we improve so the condition if we restore the function of this primary driver gene in mutant mice? So this will allow us to better understand if we, at one time, we should really act to rescue maybe more the function and alleviate or improve those animals. So that could be a one way to go for a further future treatment in human. And fortunately, as Anna mentioned, the KdVS Foundation has allowed us to go for that. So we are now generating a new allele that will express one of the mutations found in people with KdVS in exon 12. We select this one because we have another goal, is to go, if we can compare, not only compare the phenotypic outcome of this mutation to the complete knockout of cancel-1, but by making a very sophisticated genetic configuration that I could briefly introduce you, we could be able to answer the next question, which will be about how can we reduce some of the phenotype by re-expressing a functional cancel-1 after this expression of the mutant allele during development. Okay, so I hope you understand it, because I think it's quite important to be able to see whether or not we will be able to compensate the defects that has been set up by cancel-1 by restoring, for example, the expression of cancel-1 after a period of development which has been maybe altered by this type of mutation. So this is what we did. So it's a very sophisticated construct. But you have to understand that with the alleles that we are going to generate, we will have the expression first of the mutant allele. That is here at level of exon 12. So these boxes are representing exons, and the exon 12 where the serine 901 is located with a frame shift that will cause the stop of the translation of the protein. And by using a kind of trick, the same thing that I talk about, the Kree-lock system, briefly during the introduction, maybe I didn't even mention it, we will be able to put back, in fact, the normal sequence of the gene with no mutation later on. And this we can do at different type of the life. We can do it very early during development, or maybe later during the adult, to see if how far we can go by manipulating the function of cancel-1 and restoring how far we can go by restoring the function of cancel-1 at different time. So, I think this is what we are aiming at, and at the moment we are on our way to make the alleles. So we have the construct are made, and we have ES cells which are produced. So and we are waiting for the transmission. And we are, because this is quite a sophisticated construct, so we have to go through the embryonic stem cell route, and we cannot do that with a CRISPR-Cas system, for example. It's too difficult. The region is too large to manipulate. And this is ongoing at the moment, so we are here, but this is normal. I think we got the funding end of last year, and we are at month 6, so we are on time. So the next step will be, of course, to compare the outcome with a mutant allele to see if we replicate some of the defects that we observe with CONCEL1. And this is the funding that we receive from the foundation. And I will stop here. I would like just to thank those people who contributed mainly to the analysis, Thomas Borgast and Melanie Thomas. And this was done also with the help of the Mouse Clinical Institute that I'm directing here with key people. And of course, this will have never been done if we haven't met Bert, David, and Anne a long time ago, building connection and working also with Naël Nadis Kasri that you may know. So thank you very much for your attention. These are the funders that help us to make works on our models. In Strasbourg, we are working on different types of disease. But of course, our interest on KdVS is really high. So I will stop here. And if you have any question, you are welcome.
Thank you. That was great. You've done so much work phenotyping those animal models. How long did it take for you to do the experiments that you just walked us through from that initial paper? How many years of work is that?
I think it was about, okay. So making the models took us 2 years. Analyzing the models took us 3 years. And now we are pursuing it. So, you know, it's not a never-ending story, but we are looking at more precise phenotype. So
yeah.
The idea at the beginning and what we published is to demonstrate that the model is relevant for the condition. You know? This is what, because we can now start to use it to understand and to go further into that. And we also, and this model are also available to others. People can just ask and they can get it, you know, have no problem for them.
Yeah, I think there's a litter of these in Emory right now, thanks to you.
Yeah, sure.
Yeah, so no, that's. Can I ask you a couple of specifics? So I wrote down a couple of questions
sure.
that I thought of while you were showing your slides. So the first one is, and every single question is going to be couched in like, how is this helpful from a clinical perspective? So the first question would be about one of the models that you made. You made a duplication model, which, as I am aware, that's not observed in KdVS, but maybe in other conditions. And so
yeah,
how does that help us understand Koolen-de syndrome?
So for us, it was at first it was a control, you know, because when you make this type of it was a control, and it was also to explore another type of mechanisms. When you have this type of rearrangement, a deletion in the genome, you are now bringing together things which are not been together before because they are separated by the region.
Yeah.
You understand that?
Yeah.
When you bring them together, you can have new interaction which were not planned at the beginning. So saying that it's maybe not only the loss of material which is causing the disease, but maybe this new genetic configuration. Having now the model in which we have a deletion on one side and the duplication on the other, we can restore the number of copies to 2. It's artifactual, I agree. But then you can see there are still some phenotypes which are seen, and this could be due to this genetic configuration changes. Yeah. Okay, so at the beginning it was done for that, and we did that also for another type of disease. I agree with you. The duplication was not, when we did it, was not really interesting for pathophysiology or for any human disease. But now it has been shown that the duplication of this region has been found in people with neurodegenerative disorder, which is a completely different disease. So there are people who are interested in that. So it's another, we need to, so all the analysis that I showed you have been done on young adults. They have not been done along aging.
Mm-hmm.
And I think, and this is something that I took from my experience working on Down syndrome, which is one of my most important topics, in fact, in my lab, is that it's really important also to look for aging phenotypes, because for KdVS, it's maybe different because KdVS is quite new, but even in people with Down syndrome, you know, they were at long time ago, they were not able to live longer, but now they can. And now we see that they have also additional disease or comorbidities, which are appearing along aging. And with these models, we can study that. So maybe for KdVS is not the goal at the moment, but maybe one time it would be of interest to do that.
I actually thought about it from a completely different perspective when you were introducing it, and when I was reading it in your paper, is that it could also be sort of the basis upon which you might argue whether there's a dosage effect, right? Your duplication, that you'll have 3 copies of CANSL1. So when we're thinking about treatments, if you see no gross phenotype, you're less worried about.
Sure. That could be, yeah. This is another point it's completely right.
Okay.
I will be, so, but this, for this, so for the neurodegeneration, of course, there is a very strong candidate because the gene MAPTO is known to be involved in different type of dementia when it is mutated. Right. And for probably it's a key gene for this neurodegenerative effect that we have, that is attached to the duplication. This has not been demonstrated. This is only a correlation for the moment. I've never seen somebody who did that. So, but maybe it will come. We could have done that, but we need money. It's always a problem of funding, but
yeah,
okay,
I know.
we don't. So, but that's something that should be done. But I think you are right. If you don't see major changes due to the overexpression to 3 doses, that's going to be kind of a good. Yeah. I agree completely.
So we have, okay. Blake, do you want to ask your question? I don't know if you're still on, but do you want me to read it, or let me see here. Are you still on and able to read? There's a question which was a really good one.
I don't see the chat. So
okay, here, I'll read it.
Okay. Have you contacted with drugs or chemical compounds?
So I told you, everyone's thinking about, phenotyping basic research models is, we can't do that part without sort of the next part, but everybody's sort of thinking about the next part, right? So
I agree completely. So for the moment, we have not done that. I've seen some papers showing that there are some changes in some pathways in cells and the iPS cells. I've seen those papers. So we could do that. We could try those type of drugs for sure.
Yeah.
And of course, what we can do is that we can use the phenotypes that we have observed at the level of the organisms to see if the drugs are improving or reducing the phenotypes. Let's say the consequences. Yeah, this is something that is for sure something that we would be happy to do and very interested to do.
Yeah.
Yeah.
I have a question. So the most of the phenotyping is done at sort of a young adult age, right? They're somewhere between 12 and 16 weeks.
Yeah.
And so far, nothing has been, you haven't done sort of later in life to even see if there's a degenerative model, if there's any sort of worsening of phenotypes, or even when it appears, right? How early does it show up?
Yes.
One thing that I would be really interested in is, based upon the work that Bert and David have done, is it seems like there's a couple new sort of less, or I guess I'll say new clinical features. One of those is not really that new, but like sort of congenital heart defects. If those have been observed, how many of the new clinical features of KdVS are observed in your existing models?
Unfortunately, I cannot answer. We should do that. Probably So let's put it this way. I know about some other models in which there are cardiovascular defects. And usually we see a loss of, we don't see a 50-50 transmission rate. And this is what we have also with the KdVS mouse model. So on a pure genetic background, on a pure basic genetic background, we have only 30% of variability in newborn. So those animals that die before, cardiovascular defect is probably the main cause for that. Because this has been found in many other models. If you bring them into another genetic background, you always have this, what we call the hybrid dynamism, dynamic, and it helps a lot. So you get to 50-50. But this is what is observed. So I think that would be something to do. Yeah, but probably we will need to do that during embryonic development. Yeah.
Yeah. The other thing I was thinking about was one of the things that's shown up really recently in some of the work that the foundation has done is that communication deficits seem to be, I mean, they're not independent of the cognitive deficits and sort of working memory deficits that you saw in the rodent models. But is there a way to measure communication in KdVS? Like, do you do like vocalizations?
We do that. Yeah.
I mean, it seems like you have a really elegant neurobehavioral core.
No, so yeah, we do that. So we have now, so we have we can measure USV, so ultrasonic vocalization, because the mice, they speak, but in a very high level of frequency, so beyond what we can hear.
Yeah.
And there are two main levels, in fact. It's not really vocalization to exchange words. It's more about excitation and exchanging emotion, let's say. It's more about that. But it works. So we can do that. We can do that in pups. We can do that in newborns. Vocalization is better in between females and in between males. This is a basic of the mouse model. So we can do that. So we have not done that. But this is something that we can do, yeah. Because at that time when we did the first publication, it was in 2017, so almost 7 years ago now.
Wow.
We did not have those tools, but now we have, yeah. So it's a matter of reactivating the process. So now, so we got funding from the KdVS to start with this new model, with this point mutation. We can maybe take the advantage of this new course to go for other tests to see whether or not they replicate it in this specific genetic condition, and then we can go back to the deletion or to other type of models.
Wow. Well, let me see if there's any other questions. Usually I just start asking and I forget to ask people if they have. Okay. I don't see any other questions in the chat. Okay. Well, I am going to, so thank you very much for your time and walking through all of that work. I can think of about 1,000 different new projects that I think we need to figure out, and questions that we can use your models to address, especially the sort of screening compounds or like promising things that we think could be.
I think we should figure that for a very late phase of the screening of the compounds, but I think that would be something.
Yes, absolutely.
Yeah, for sure.
I'm going to stop the recording now, but stay on for just a second.
Thank you very much.
Yeah.
It was a pleasure.